Okay, so today we are going to learn about Kreb cycle, which is the third stage of aerobic respiration. And it uses the product which has been formed in the link reaction, that is acetyl coenzyme A. So this compound which has been formed in the link reaction, it undergoes a series of cycles that is known as Kreb cycle. It is a closed pathway that is regulated by enzymes. Now here we don't need to memorize each and every enzyme that is involved. We just have to know what substrates are being formed in the whole cycle, and how many molecules of NAD and FAD have been produced, and more importantly, how many molecules of adenosine triphosphate, that is ATP, has been formed, because our main purpose is to know perform this cycle is to generate energy-rich molecules in order so that it can be used. So here is a short quick recap of the link reaction which we studied earlier. That pyruvate molecule, which was a 3-carbon compound, it forms acetyl coenzyme A. Now this acetyl coenzyme A undergoes Krebs cycle. What's happening here is that this acetyl coenzyme A, first of all, in the first process, it gets combined basically. It gets combined with oxaloacetate, which is a 4-carbon compound. So these two compounds, which are 2-carbon compound acetyl coenzyme A and 4-carbon compound oxaloacetate. By removal of this coenzyme A, the main function of this coenzyme A was just to carry this acetyl group into the Krebs cycle. So what happened here is that this acetyl coenzyme A reacts, it combines with the 4-carbon compound oxaloacetate, and it forms another 6-carbon compound, that is citrate. So as the first product of the cycle is citrate, this cycle, the Krebs cycle, is also known as citric acid cycle. Okay, so the first product form is citrate, which is a 6-carbon compound. Now what's happened here is that this citrate, it undergoes dehydration, that is removal of a water molecule. This takes place and it gets converted into another molecule named as cis-aconitate. This is also a 6-carbon compound. This water which has been removed in the first step, it gets added with this cis-aconitate. That is, the second step is hydration of cis-aconitate. Hydration of cis-aconitate takes place, which forms another compound named isocitrate, which is also a 6-carbon compound. Then isocitrate, it undergoes, as we can see here clearly, that a hydrogen carrier molecule NAD, it is getting reduced. Now, when does it get reduced? When the compound, the substrate, undergoes dehydrogenation, removal of a hydrogen molecule. This hydrogen combines with the NAD and it forms reduced NADH2. Now, in the next step, it forms oxalosuccinate, which is also a 6-carbon compound. So isocitrate undergoes a dehydrogenation. So third step is dehydrogenation of isocitrate. Now what happens next is this oxalosuccinate, it undergoes decarboxylation, that is removal of carbon dioxide, and it forms another compound. Now, important thing to know here is that this is a 6-carbon compound. It is removing its 1 carbon atom in the form of carbon dioxide. So when we reduce 6 carbon atoms by removing 1 carbon atom, we get a compound which is 5-carbon containing compound. So oxalosuccinate, which is a 6-carbon containing compound, it removes 1 carbon atom in the form of carbon dioxide. This process is called decarboxylation, and it forms a compound which is 5-carbon containing compound. This is named as alpha ketoglutarate. This alpha ketoglutarate now undergoes a series of changes, which is first it removes a carbon atom in the form of carbon dioxide. So alpha ketoglutarate undergoes decarboxylation. Now the coenzyme A, which was removed in the very first step of Krebs cycle, by removing coenzyme A and the addition of acetyl group and oxalosuccinate, we formed citrate. So here in this step, this coenzyme A gets added. So second thing which happens to this alpha ketoglutarate is addition of coenzyme A. Now next what happens is the third thing, dehydrogenation, removal of a hydrogen molecule. This hydrogen molecule gets attached to the nicotine NAD basically, and this NAD gets in its reduced form, that is NADH2. So three things happens to the alpha ketoglutarate, that is decarboxylation, removal of 1 carbon dioxide, then addition of a coenzyme A, and lastly removal of hydrogen, which gets attached to the NAD, forming reduced NAD. Okay. After the series of changes, what happens is this alpha ketoglutarate molecule, it gets changed to succinyl coenzyme A, which is a 4-carbon containing compound. As we can see here, that alpha ketoglutarate was a 5-carbon containing compound, but due to removal of 1 carbon atom in the form of carbon dioxide, it formed the compound which is 4-carbon containing compound, succinyl coenzyme A. What happens to the succinyl coenzyme A is that here we can see we can form our first energy-rich molecule, adenosine triphosphate. So what's happening here is that removal removal of an inorganic phosphate from succinyl coenzyme A. This inorganic phosphate gets attached to the ADP and it forms adenosine triphosphate. So we can have our first molecule of energy being formed here. Now this succinyl coenzyme A, it gets converted to succinate, which is also a 4-carbon compound. By formation, in this step we can write as formation of ATP. Then what we have is this succinate, which is a 4-carbon containing compound. It undergoes dehydrogenation. Dehydrogenation, this hydrogen now gets attached, that gets combined with another hydrogen carrier molecule, which is flavin adenosine dinucleotide. This is a new hydrogen carrier compound, which gets reduced into its reduced form FADH2 in Kreb cycle only. So this hydrogen, it gets combined with the FAD and it forms its reduced form FADH2, forming a compound called fumarate, which is a 4-carbon containing compound. So what's happening here is this succinate, it undergoes dehydrogenation. Next, what happens here is that fumarate, which is a 4-carbon containing compound, it undergoes molecular rearrangement and form malate. Then last step is that this malate, which is a 4-carbon containing compound, it undergoes dehydrogenation. This hydrogen gets attached with the NAD and forming its reduced form NADH2. And the substrate being formed is oxaloacetate. Now when another molecule of acetyl coenzyme A comes to this cycle, this oxaloacetate can be reused to get combined with the acetyl group and then follow the cycle again. So the whole process can be repeated again. So in each cycle, what we produced is we had 2 molecules of carbon dioxide being formed, 1 molecule of FAD being formed, which is in the reduced form obviously, and then 3 molecules of NAD formed, which is in the also in the reduced form, and 1 molecule of ATP being formed in the whole process of Krebs cycle. So basically the link reaction is also considered to be a part of Krebs cycle. So in that process, we remove 1 carbon dioxide molecule, which is also counted in the Krebs cycle. So we can write it as 3 carbon dioxide molecule being formed or removed in the whole process. Because these 3 carbon dioxide molecules are formed in the Krebs cycle, it is also known as tricarboxylic acid cycle. Cycle.